Okay, first off, couple reminders here, guys. Your cell anatomy lab report is due on October 24th. That is Thursday, okay, of this week. So make sure that you have it done, okay, for October 24th. Okay, so you don't take any late penalties or anything on that. Make sure that you watch the podcast when you put it together, and as well, look at your microscopy lab, okay, especially your lab drawings. Okay, if you didn't get full marks, make sure you do it a little bit differently this time, all right, so that you get as high a mark as possible there before the weekend. What we're going to go over today, guys, is going to involve us reviewing a couple of things from last week, okay, and then talking about kind of their implications. First off, the big three methods of cellular transport. Anybody remember what they were? Right. Okay, we had osmosis, we had diffusion, and active transport. Yes, you should write this down. All right. Now remember, we said we had to be able to identify these from a situational type of question on an exam, all right, where I would say, here's what's going on, identify what the main cellular transport process would be, okay, or how it would be affected in this situation or something along those lines. The good news is that each one of these has at least one or two unique characteristics compared to the others, all right, so they should be easy to identify. All right, what is osmosis? Okay, on the right track. The big thing about osmosis is it's only going to transport water. Okay, so that's probably, okay, your defining characteristic for osmosis, all right, is that it is only transport of water. Okay, now Kean also mentioned it moves from high to low. To some extent, yes, okay, but it's moving across what in osmosis? Not across the cell, but across the membrane. Okay, it's water moving across the membrane. Okay, and what's it trying to balance? Right? Right. You bet. We'll say solutes because that's probably more accurate than saying salt. Okay, it's usually an ionic compound, but okay, to balance solute concentrations. Okay, does it take any energy? Or is it passive? Passive, right? Okay. In fact, the cell has no control over it whatsoever. It simply happens. All right, which can be good or bad. Okay, we talked about that. If we drank a bunch of salt water, the fact that we can't control osmosis would work against us because our cells would shrivel up. Okay, water would be automatically drawn out of them to help flush out and balance the salts that we had taken in. Okay. Question? No? Okay, and this is one of the reasons why you don't want to have a whole bunch of salt really right before you have kind of a big competition, okay, or something like that if it's athletic, because it's going to draw water out of your muscles, which could result in your muscles possibly getting cramps, fatigue, things like that. If you drank too much distilled water, it doesn't really work the same way. Yes, water would move into your cells and you might retain some water, gain some water weight, okay, but in general, your kidneys will get it out for you. Okay, I mean, if you've ever like drank a big Slurpee, right? I mean, it's not long before you're headed to the bathroom, right? And it's not just because it probably has caffeine in it, but also because you've just ingested this huge amount of fluid, and your body's going, well, we can't hold all of that in here. Let's get rid of some of that. We don't need it all, right? Yeah, that's old school thinking. Yeah. Well, there are electrolytes in salt, right? But not to that extent. You don't need to just take a handful of salt, right? Yeah, but again, that would have been, again, working against them. Oh, let's eat some salt and not give you any water, right? It's like, yeah, that's, yeah, recipe for heat exhaustion, recipe for, yeah, right? I mean, and people have, and this was the big problem in football in the States, in a lot of places, guys were dying, okay? Literally dying in practice because they got heat exhaustion. right? To the point where their heart stopped. Right. So yeah, you have to be really careful about that kind of stuff. Osmosis, though, is passive, and your cell has no control over it. Okay. Key points: it's water. It's moving across the membrane to balance solutes. Okay. It's always across the membrane. Now, with diffusion, can diffusion transport anything? Yes. Okay. So moves anything. Okay, but it's within the cell. All right, it's not moving across the membrane. It can move when it's outside of the cell. Things can move by diffusion outside of the cell, but it doesn't move across the membrane by diffusion. Okay, so moves anything within the cell. Okay, what else? It's passive. Yep. Which way does it move? Right. Okay, moves everything from high to low. It's that natural sort of dilution or dispersion of things. Okay. Any drawbacks? Right, it's slow and not targeted. Okay, we can't ensure it gets to a certain particular point within the cell. It will likely get there because if it's needed, it's probably an area where it's low concentration, right? And it'll work its way there simply because that's the way things move, right? But again, it is slow, all right? And that is, okay, kind of the big drawback of diffusion. It's slow over large distances. Is the distance inside of a normal-sized cell very far? No. Okay, so it works just fine because your cells are microscopic and diffusion works great over microscopic distances. All right, active transport. One of the key points is in its name. Right, it's active, okay? It uses energy. Okay, and because it's using energy, it can do the opposite of diffusion. It can move things from low to high, okay? So we call that against the gradient. And it also works across the membrane. We have to remember that. Okay? Active transport is across the membrane, not within the cell. Okay? The only way to move things within the cell, okay, within the cytoplasm of the cell, is diffusion. So active transport uses energy, moves things against the gradient, okay, so from low to high across the membrane. Okay? And in that case, obviously, you get what you pay for. Okay? If you're spending energy, it works well. All right. Okay, so that's the quick review of the big three. Okay, remember there are going to be questions that ask you about that. Okay, today we're going to be looking at the effect of cell size on intracellular transport. All right, intracellular means within the cell. So which of the three are we looking at? Diffusion, right? Okay, this is the big one that goes along with what we're talking about today. Okay, so we just finished saying that within a cell, diffusion is how we transport things. So let's say I'm comparing two cells. Okay, this is just a big cell, okay, on the left. Nucleus is the same size as it is in the small cell, right? We got our various organelles in places throughout the cell. And I'm just going to put X's to represent places where resources are available. How many did I put in there? 6. Okay, and I'm going to use O's to represent, well, I better not use O's, I'll use squares, okay, to represent where resources are needed. Okay, so X's is where they are, squares is where they're needed. What are you noticing here right away? Right. In a small cell, things don't have to go very far. All right, which is good because we've already said the main mode of transport, diffusion, is slow. So if a cell gets really, really big, what kind of problems is it going to start to encounter? Yeah, not just slows down, but if it's transporting stuff it needs, maybe it doesn't get there in time. All right, like let's say it's needing fuel, it needs sugar at the mitochondria, but the cell is getting so big that it's taking too long for that material to get to the mitochondria and the cell dies before it gets there. Is that possible? Yeah, it certainly is. All right, this is very much like what happens when a city grows. Okay, I mean, if you go back even, well, let's go back a long way. Let's go back 30 years. Okay, and you look at Calgary 30 years ago. You could drive from one end of Calgary to the other, okay, in no time at all. All right, because Calgary was a lot smaller. There wasn't as much traffic, and there wasn't as much of anything else, so you could get through it quick. All right, as Calgary grew, your ability to drive through it decreased. Okay, took longer and longer and longer. So what did we end up having to do in order to make that better? Right. Okay, built Deerfoot. Right, Deerfoot Trail, built the extension. Right, about 10 years ago, built the Deerfoot extension. That meant that you didn't always have to take McLeod to get to Okotoks. Before that was the only way. Right, Deerfoot only ran to about Southland or Anderson, and then you had to come back in, take McLeod with all those lights. Took forever. So you built the bypass. Well, that helped for a while, but right in the last 5 years, how much do you enjoy driving on Deerfoot? I personally hate it. Okay, because if you're trying to travel on the Deerfoot during rush hour, okay, you know that you are going to be in a parking lot with a bunch of idiots, okay, for an hour and a half minimum. Okay, sometimes it's even more than that. So that's not a great situation, and that's the situation encountered by a cell that's really, really big. Okay, unfortunately, a cell can't build bypass roads. Okay, it can't build Stony Trail Ring Road, right? That things can go around the city and get out quickly, right? They just don't have the ability to do that. They have to rely on that things have to move by diffusion from where they are available to where they are required. Okay, now where do the resources that are here represented by X's actually come from? Outside the cell. So not only do they have to get maybe from here where the cell actually has a supply of them to here, but it may have actually had to transport them first through the membrane, then to here, and then over to where they're needed. All right? And the bigger the cell gets, the longer that's going to take. And it's not just about moving materials we need in. It's also about moving things we don't want in out. All right? If your cell has, been undergoing some metabolic processes, really having to work really, really hard, toxic materials build up. Lactic acid, carbon dioxide, okay, things like that. Those things are toxic to the cell. We don't want those building up. All right? And if it takes a long time for things to move from where they're produced to get out of the cell, that's a lot more time where those toxins are in contact with cellular material, and they'll damage it. The longer they stay in here, the worse that is for the cell. Everybody follow? Okay? For an organism as big as us, we can't rely on diffusion to move something around our body. It would take far too long, which is why we have a well-developed circulatory system, okay, that pumps blood. All right? Yes, to get things out of the blood and into our cells, we still rely on diffusion. That's fine. When it gets there, we're talking about small distances. Okay? Out of the blood vessel and into the cells is a small distance. We can handle that. Right? But relying on diffusion to get food from your small intestine to your brain, okay, is not an effective way to transport material. Everybody with me on that? Okay, so there's sort of an upper limit to the size a cell can achieve before it starts to be detrimental to it. All right? So if you want to get bigger and not have this transport problem, you can't stay single-celled, can you? Yeah. Okay? If you become multicellular, do you fix that problem? Yeah, because now instead of having one giant cell, you might have a whole bunch of small ones. Okay? Is that going to improve the situation for you? Yeah, because now in each cell, stuff only has to go a small distance. Now, if it needs to be transported across all of the cells, well, then you still have some trouble. All right? But generally, you don't need to do that. Cells can kind of support themselves. And in a multicellular organism, in between all of those cells would be capillaries and stuff like that that would be supplying all of those nutrients. So early on in the history of the Earth, this was the problem that was encountered. Bigger was better. Okay? You wanted to be big, but it only worked to sort of a fixed value. All right? The way to continue to get bigger was to become multicellular. All right? And when multicellular organisms first appeared, variation, kind of the amount of different kinds of life forms on Earth just exploded, right? Because it was just such a huge advantage to be able to be multicellular and get specialized and be able to move around and be big and not have this problem. Okay, it was huge. All right. So here's what we need to understand. And where do I want to? Okay. So I need you guys to write down these things here. Okay. Surface area to volume ratio and cell functioning. Okay, cells absorb nutrients and excrete wastes through their membrane. Okay, the membrane is the surface area of the cell. Okay, so essentially what we're saying here, guys, is this. The cell membrane represents the surface area of the cell. Everything has to get into the cell that way. How many dimensions does surface area have? 2. Length and width, right? If you're calculating the area of something, you go length times width. Agreed? All right. Now, that's why it's usually measured in square centimeters, meters, whatever the units are that you're dealing with. All right. Volume, on the other hand, is calculated by going length times width times height. All right. And its units are always in whatever cubed. Okay. So as a cell gets bigger, its surface area increases. It has to, because it's always encompassing an exponentially greater volume. All right. If I double the surface area of a cell, all right, the volume is far more than doubled. Okay, it's much bigger than that because we're talking about 3 dimensions now. All right, that's what creates the problem for a cell. As it grows, the area it has to take in and get things out becomes so much smaller by comparison to the volume that it has to supply. All right, so it's just like the example we used with Calgary. All right, if you got more and more people sharing the same number of roads, you get congestion. Okay, things slow down. People aren't able to get in and out as fast. All right, and that creates a problem. All right, the cell has the same problem. As it gets more and more volume, there's less and less ways for it to get things in and out. Okay, and so it slows down to the point where it may not function anymore. Okay, so that's essentially what all of this is saying. Now, from a mathematical point of view, that looks something like this. All right, let's imagine that the cell is a cube. Okay, and let's say that it is 10 micrometers by 10 micrometers by 10 micrometers. All right, just to have easy numbers to deal with for math. All right, how do I calculate the surface area of a 3-dimensional box? Length times width times height. Well, they're all 10, so 10 times 10 times 10 is 1,000. All right, so the surface, or the volume, sorry, of that cube is 1,000 cubic micrometers. All right, the surface area, though, by comparison, okay, is going to be length times width times the number of sides. All right, so 10 by 10 is 100 times 6 is 600. All right, so we've got a surface area of 600 micrometers, a volume of 1,000. That means the ratio is 0.6 to 1. Okay, is everybody with me there? All right. What I don't want is for this to get really out of hand. If my volume gets really big, okay, this ratio gets in favor of volume really fast. And that's going to be bad for the cell. All right, so let's say we make this cube a little bit bigger. Let's say that this cell grows to twice its previous size. All right, so now it's a cube that is going to tax Mr. Coder's artistic ability. All right, that's vaguely a cube. Okay, 20 by 20 by 20. Okay, I know it's missing something. It always ends up missing something when I draw 3-dimensional. I know it doesn't look right. Thanks, Jace. I appreciate your feedback. Now be quiet. I don't have to be an art critic in here. All right, so 20 by 20 by 20. Right? What do we got? 20 times 20 is 400 times 20 more. 2,400, right? Yeah. Okay. So we have a volume of 2,400 micrometers. Is that quite a bit bigger than it was previously? And all I did was double each side, right? But that more than doubled the volume, didn't it? Okay. Now, if I want to find the surface area, I'm going to go 20 by 20, right? So 400 times 6. Okay, so 400 times 6 should give me, 4 times 6 is 24. No, so this number isn't right then. Okay, give me 20 by 20 by 20. Someone punch it into there. Never mind. My math is just rotten today. I'm thinking 8,000, but I want to be sure. Come on. And I got nothing. Okay, I need someone to punch it in for me. Let me do that. 8,000. All right, so the volume's 8,000. Good. So we doubled one side, and we got 8 times the volume. Okay, so the cell is now 8 times in volume, and the surface area is going to be 2,400, right? All right, 2,400 to 8,000. Is that getting worse in terms of volume? Yeah. Okay, so can someone punch in 8,000 divided by 2,400? 4. Okay. No, it should be 3. Yeah, it should be 3. 1 to 3. All right. So we got 1 to 3, whereas before the ratio was 0.6 to 1. This is a far better ratio for a cell to work on because the surface area and the volume are far closer than they are here. The volume is 3 times bigger than the surface area here. That is not a situation that's going to be good for that cell. The bigger it gets, the more problems it's going to have using diffusion as a way to move stuff around the cell. Okay, is everybody following me there? All right, so what we're going to do today is I'm going to give you a handout here. It's got an activity on it where you're going to have to do a little bit of calculating volume and surface area, just like I demonstrated here, okay, with cubes of different sizes. One of those cubes, however, is going to represent a multicellular organism. All right, so it's going to be a cube that's full of small cubes. All right, is its volume still going to be the same as a big cube of the same size? Yeah, because volume is length times width times height. But if you're made up of a whole bunch of tiny cubes, each one of those cubes has 6 sides. What's happened to your surface area? It's way bigger. All right. In fact, your surface area to volume ratio will be the same as a small cell. Because what are you made of? A whole bunch of small cells, each one with the right surface area to volume ratio. So again, there's the advantage of being multicellular. You don't have this surface area to volume problem because you're made up of a whole bunch of cells that are the right size. Okay, is that making sense? All right, so I'll hand those out and then we'll talk a little bit more about what you got to do. Okay, so listen up here, guys. First thing, okay, some sizes and stuff on there. Okay, just add these sizes because for some reason these numbers that I thought I had erased really showed up well when I scanned them. Like they're invisible on my original, but they showed up on the scan for some reason. Ignore those hundreds that are on there. Okay, the size of micro is 1 by 1 by 1. Okay, so each side is 1, let's say, micrometer. All right, macro is 10 by 10 by 10, and so is multi-micro. Okay, it is also 10 by 10 by 10. Okay, one of the things you're going to be asked to do is fill in a chart like this one, okay, for those cubes. Micrometers, yeah. Okay, so if I want to calculate the surface area of any one of these cubes, I go length times width times number of sides. Okay, if I want to find the surface area, it's length times width times the number of sides. Okay, that'll give me the surface area for each one. Okay, for volume, then I go length times width times height. Okay, and then I write in the ratio here, all right, of the surface area to volume. Okay, and making sure that your surface area is number is 1. Okay, so it's 1 to something. Essentially, then you're telling me how many times bigger, okay, the volume is than the surface area. All right, the closer those numbers are together, the better that cell is, okay, in terms of functioning. Okay. Now, for some reason, I don't have the first part of that file in here anywhere. Okay, listen up here, guys. So, same as always, this is kind of like a lab, except it's a little bit less work. All right, you don't have all of the stuff in that you would have in a regular lab report. All right, there's no there's no variables. Okay, no design here. There's also no evaluation because there's no places where there can be a source of error in here. You're not actually doing an experiment. All right, so we just need to have a hypothesis here that is an if and then statement about the relationship between surface area and volume and the functioning of a cell, all right, which is what we've been talking about since the beginning of class. So you ought to be able to make an if and then statement, the if part talking about cells functioning and what they have to transport and how they do it, okay, and a then part that talks about what happens as a cell gets bigger. Okay, everybody follow me there? All right. Now, the procedure is as follows. You need a 3-column table that looks like the one I had up here just a minute ago. This one, okay? All right, so you're going to have the cube names, micro, macro, multi-micro, okay, your surface area, volume, and surface area to volume ratio. Okay, I'll put that back up in a minute here, okay? I'll put it up in a minute here, okay? And what you need to do is calculate the surface area and volume of each cube, which we already gave you the dimensions of, okay? We told you that micro is 1 by 1 by 1, and macro and multi-micro are 10 by 10 by 10. Now, the thing to remember here about multi-micro, it's filled up with tons of little cells, the same size as micro. How many do you suppose are in that cube? 10 times 10 times 10. How many is that? That's 1,000. So, if you know the surface area of micro, the surface area of multi-micro is how many times bigger than that? 1,000 times. Okay, micro's 1 by 1 by 1, yes. Okay, everybody with me there? So I've given you a big juicy hint there. You shouldn't screw up the calculations now. Okay, all right. Once you've got that done, okay, then you've got the analysis here, and there's a few questions to answer. Okay, the first one is basically just asking what happens to the dimensions of the cube when it gets bigger. Okay, the second one is talking about the two big cubes. Okay, they're the same size, but one has a bigger surface area. To calculate how many times bigger the surface area is of multi-micro than it is of macro. All right, that's the kind of the big thing, is the advantage of being multicellular over being one big giant cell. Okay, for number 3, for which of the two cubes does the ratio between volume and surface area change when the size increases? All right, so now you're looking at all 3 cubes. Here's the thing to remember though. If you make multi-micro bigger, do the little ones inside get bigger? No, it just fills up with more of them. Okay, so that's the thing you got to consider here, because otherwise sometimes people get the wrong answer. All right, if you're a multicellular organism, when you grow by making more cells, not by making your cells bigger. Everybody follow? Okay, so your surface area to volume ratio kind of stays the same. Oops. Okay, number 4. Use the diagram below right to answer this question. So that is this one here. Okay, these cells, because that's what they represent, okay, all have the same volume, but they all have different surface areas. All right, now when you do the calculations, you'll actually have to measure. All right, they'll come out pretty close. When you actually do the measurements, the volume ends up being slightly different, but very close to the same. But the surface areas are way different. All right, so what this question is asking about is, does the shape of a cell have an effect on its surface area to volume ratio? Can a cell change its shape to improve its surface area to volume ratio? All right, what you need is to have the mathematical proof, yes or no. Okay, so you calculate the surface area and volume of each of those, okay, and then give me the surface area to volume ratios. Now remember, these aren't cubes anymore, okay? They're slightly elongated. So what you'll have to do is figure out this side and multiply its surface area by 2, because there's 2 of it, one on the front, one on the back. Okay, figure out these, multiply it by 4. All right, for this one here, you've got 2 of each type of side, so 2 of this side. Okay, so 2 of this side, 2 of that side, 2 of that side, add them all together. All right, and same for this one here, 2 of the long skinny side, 2 of the long wide side, okay, and 2 of the front. Okay, add them all together. Okay, and then the volume is just length times width times height for all of them. For number 5, it's kind of a, all right, you've done this with these cubes. Now let's look at some cells within the body that have similar types of dimensions. A liver cell is kind of like A. Okay? A skin cell, which you see here, is kind of like B. And a nerve cell, which, believe it or not, in your spine can be up to 1 meter long. Okay? Still microscopic, but 1 meter in length. All right? Is kind of like C. All right? Yet they all still function because they still all have a good surface area to volume ratio. So you kind of have to look at what you found in number 4 and use it to answer question number 5. Okay, everybody follow me there? All right, then your last 2, which showed up where? Didn't show up. Oh, I pasted, that's right underneath. Okay, 6, okay, is showing red blood cells. You looked at these in the lab. They have that kind of donut shape. All right, they have that donut shape because they dip in the middle where there's no nucleus. Does that dip in the middle affect its surface area to volume ratio? Okay, so that's what number 6 is asking. How is its shape beneficial for its job of carrying oxygen molecules attached to the cell membrane? All right, does having that shape make it better, worse? Okay, what's the case there? And then number 7 is kind of the gist of the whole thing. All right, most materials get from place to place in the cell by diffusion. What effect is a low surface area to volume ratio? Okay, so low surface area and high volume, that's the bad situation, right? Going to have on intracellular transport. All right, so there's a fair amount of questions to answer here. All right, and then your conclusion, based on your calculations here with all the different cubes and cells, okay, does it hold true? And that's it. Okay, everybody follow there. Okay, you've got about 40 minutes of class time to work on that. Okay, you should be able to get a pretty good dent into that, I would think, in that amount of time. All right, I'm going to look at the calendar here and figure out when I'm going to make this due. All right. Yes. Okay, guys, so there's been a little bit of confusion. I think it's partly my fault here on calculating the surface area to volume ratio. So let's just look at micro as an example here. Okay, the surface area of micro is 1 times 6. So its surface area is 6 micrometers square. All right. The volume of micro is 1 times 1 times 1, which is 1 micrometer cubed. Okay, so the surface area to volume ratio of micro is 6 to 1. Everybody follow me there? That's how we do the ratio, right? Okay, now if I'm looking at macro, right, and I'm going to kind of give you the answers to macro here as well, okay, but I'm sure most of you are that far. Okay, macro's surface area, okay, is 10 times 10 times 6, which is 600 micrometers square. Whereas its volume is 10 times 10 times 10, which is 1,000 micrometers cubed. Everybody with me on that? All right. Now that is not 6 to 1, but 0.6 to 1. Right? Because they can both be divided by 1,000. Everybody with me there? Okay, so that's what we're looking for in terms of doing that calculation. I'll leave multi-micro to you guys because we already talked about how to calculate its surface area and its volume. Okay, but that's what we're looking for in terms of numbers. Okay.